because there wouldn't be enough density in that. There could be 2 or possibly 3. There was even a 4 spiral model put together. But the thing about the Watson-Crick model is that it's just right. It's one of those things when people looked at it, it looked perfect, and it looked so perfect that people wondered why they hadn't thought of it themselves before. So for this structure to work, it's got to make sense. And that means that chemically and physically, it's got to hang together. It's got to be a stable structure that won't be tempted to fall apart or disintegrate at the wrong time, because it's got to have a series of robust functions attached to it. So structurally, it's got to make sense. And that's exactly what this structure did. You've got these 2 ribbon-shaped things going down. These are the sugar phosphate backbones, very monotonous repeating structure with no variability at all. And they are held together in this double helix, always exactly the same distance apart, rather like the handrails going down a rather fancy looking spiral staircase. And the reason that these 2 ribbons are always the same distance apart is that the steps between them are always of identical width. And if they weren't of identical width, then the structure wouldn't actually work molecularly, and it wouldn't look nearly so beautiful. And the other thing about this structure, it was recognized by this time that DNA was the stuff of genes. And again, that wasn't something that was intuitive when it was first discovered. But somewhere in this structure, you've got to be able to spell out the billions of instructions that make up the directory of life, if you like. So somewhere in there has got to be the capacity to spell out the alphabet of the genes. And they pointed out that the sequence, if you take one of these strands, say the left-hand one there, and look at the letters that are marching up there, you've got A, C, T, G, and then it turns the corner, it carries on A, T, G, G. And the sequence of genetic information, they argued, was embedded in the sequence of those letters along the length of the DNA spiral. So certainly in there, you've got the capacity to spell out the genes, and I'll explain what A, T, C, and G are in just a moment. And the other great thing about this molecule, if the structure is correct, is that somewhere in there, the molecule must have the capacity, the ability to be able to copy itself. Because when cells divide, the chromosomes split up the middle, the DNA replicates, and you end up with 2 daughter nuclei containing exactly the same genetic information. So somewhere in there, you must have the capacity to be able to copy the genetic information that is encoded in DNA. So the structure must be able to copy itself. So what Watson and Crick did at the heart of their deduction was to look at the steps, the base pair steps that link these 2 ribbon-shaped handrails. And again, the ribbon-shaped handrails, there's nothing remarkable in there. It's just sugar phosphate alternating all the way to infinity. So there's no capacity to carry variable information in there. So the variability resides automatically in these cross steps. And each step is made up of 2 unequal halves. Now these are the bases, and the slightly bigger bases are the A and the G, and the slightly shorter bases are the T and the C. And what Watson and Crick worked out is that it sounds very straightforward, but A goes with T and C goes with G. And that's why the steps end up exactly the same length. If you have got a left-hand bit, as at the top of the diagram there, that's an A, that's an adenine, then it automatically connects with a thymine, a T. If you've got a guanine, the G, then it automatically attaches to a cytosine. And if the T is on the other side, then the A is its partner. If the C is on the other side, then G is its partner. So A always goes with T and C goes with G. And that is the basis of their discovery. This explained at one stroke how you end up with steps that are exactly the same width, so that holds the helix in shape, and also how you can accommodate an infinitely variable progression of these letters along one strand and still end up with the steps the same width, because they pair automatically with their partner. They know which partner they've got to pair with. So here's an example of an A, an adenine, pairing with a thymine, a T. And by the way, we're moving beyond the biochemistry in about 2 minutes. So this is the end of the biochemical bit. And then we get on to the personalities and the other things, which are possibly what you want to hear in the talk. But just to explain how the A and the T as an example of the base pairs hang together. So here we're looking down effectively on one of these cross steps in the spiral staircase. And we've got the adenine on the left, and it's the bigger half. It's got 2 rings in it. The thymine on the right is the smaller half. It's only got 1 ring. And the dotted lines that hold the 2 halves together to make up the full step are what are called hydrogen bonds. And again, this is one important bit of biochemistry, because these are the bonds that are quite firm, but not rock solid. So if you like to think of them as a handshake, then you can release the handshake. And when the DNA molecule splits up the middle to replicate itself, and these hydrogen bonds are severed and the 2 halves just move apart. That's distinct from the covalent bonds, which hold the other structures together. And they're not so much a hand clasp as a pair of handcuffs, really. So the hydrogen bonds are what give you the A pairing with the T and the C pairing with the G to give those steps of exactly the same width. Now, their second throwaway line comes towards the end of their paper. It's a very short paper, by the way. It's only 800 words or so. And it says, it's not escaped our notice that the specific pairing, in other words, A with T and C with G, we've postulated, immediately suggests a possible copying mechanism for the genetic material. And this really is the basis of the whole of the molecular biological revolution. It goes right through to all the fancy PCR tests we're seeing in the COVID pandemic and all the rest of it. And they elaborated this idea in this diagram. And what you see here is a stretch of the double helix. That's it, intact at the top of the picture. And then it has split down the middle, and it is reassembling its partner strand as it goes along. And the new strands you see there. And if you look at the old strands, first of all, if you take the old strand on the left, you've got the sequence TTA. And because A goes with T and C goes with G, then that automatically means that you have AAT lining up to go with TTA. And that's exactly the same as it was on the old strand that was partnering it before. So what you're doing here is simply because you've got one set of letters up each of the strands, those letters know automatically which partner bases they're going to pair with in order to recreate the missing strand and so duplicate the whole double-stranded molecule. So that, in essence, is what the excitement of the discovery was. They came up with a coherent structure. They showed that it worked biologically. It had the capacity to work biologically in encoding the genes, and it had the capacity to replicate itself as well. So that's what Watson and Crick did. So here's the beginning of a list of things that Watson and Crick did not do. And again, when I started rereading the double helix, there were some things that I'd missed the first time around, because the first time I read it, I was ready to start medical school. So there was lots of stuff going over my head then. If you read it, you rather are left with the impression that actually Watson and Crick discovered DNA. Well, they didn't. DNA was discovered actually in the 1870s by this guy who was a Swiss man called Friedrich Miescher, working in Tübingen in Germany, in one of the very first of the biochemical labs. And he was given an experiment to pull out new proteins, which were all the rage at the time, from white blood cells. And he worked out that the best source of these was actually pus-soaked bandages. So he worked in the biochemical department in the castle high on the hill in Tübingen. Every morning he would toddle down the hill with a bucket to the surgical wards in the hospital, where he'd fill up the bucket with these slimy pus-soaked bandages. He'd trek back up the hill. He'd then spend days and actually weeks pulling out the single white blood cells, neutrophils from the pus, and then rupturing those and popping out the nuclei. So it was a little bit like trying to pit microscopic cherries on an absolutely tiny scale. And his project was to try and find new proteins in the nucleus of white blood cells. And he found a couple of those, and then he found an extraordinary chemical, an extraordinary substance that didn't behave at all like a protein. And he got so fascinated by it that he abandoned his protein research completely and then concentrated on this new stuff. And he called this new discovery nuclein because it came from the nucleus. It was acidic. He managed to work out a chemical formula using very laborious old style techniques. And that's his attempt at the chemical formula there. And when you add up all those atomic weights, you end up with a molecular weight that comes in at just under 1,000. So it's quite a big molecule, but it's nowhere near as big as proteins, which are tens of thousands molecular weight. He did quite a lot with his nuclein, but Misha could never quite work out what it was doing. And he found it in all sorts of places, in salmon sperm as well as white blood cells. He really managed to dig it out from a variety of places. And towards the end of his life, he was writing letters to his great mentor. And at the time, he was actually dying of tuberculosis in a sanatorium in Davos, up the Swiss Alps. And some of these letters make very poignant reading indeed. But one of his last letters, his mentor writes and says, "Tell me, Friedrich, what do you think your discovery nuclein actually does?" Because there is a rumor that the new structures that people are discovering in cells, which people are now calling chromosomes, consist of a material called chromatin. And it seems that chromatin is rumored to be very similar, if not identical, to your nuclein. So do you think that your nuclein from the nucleus could have anything to do with the chromosomes, which are thought, we're beginning to think then, are the basis of heredity? And he wrote back and said, "I still don't know what nuclein does. I think it's very interesting. It might be a store for phosphorus in the nucleus. And I think it's completely irrelevant to inheritance and genetics." So with that phrase, he wrote off his discovery as something completely irrelevant to what we now understand to be the main function of the nucleus. Here's the second thing that Watson and Crick did not do. They did not prove that genes are made of DNA. And again, if you read the book, then you're sort of left with that impression. But actually, genes are made of DNA was the work of two people. First on the left is Fred Griffith, who was a British bacteriologist, a microbiologist, worked in a public health laboratory in Covent Garden, and he was an expert on the pneumococcus and various other infectious germs. And his work was picked up, and I'll explain what he did in just a second. His work was picked up later by an amazing man called Oswald Avery, who was also a pneumococcus freak, but he worked on the other side of the pond. He was at the Rockefeller Institute in New York. And again, of all the people in the DNA story, you know, every time I research one of these things, I work out who is the person that I would really most like to spend an evening with. And it certainly is not Watson, it's not Crick, could be Maurice Wilkins, I think. But Fred Griffith, I think I'd be very interested to meet. And Oswald Avery, actually, he's the man that to me would take the prize. And he was very reclusive in his way. He retired to his laboratory each day and he refused to be disturbed. You could hear him whistling tunes from Wagner operas through the closed door. He just got on with his work. And he had a way of introducing people to a new research topic. So bright young things wanted to come and work in his lab in New York, and he would get them in. He wouldn't give them any idea what they would be doing at all until one evening when he would corner them in the lab. He would sit them down and he'd subject them to a 2-hour monologue on the pneumococcus, his favorite germ, and where he thought that the growing points of knowledge might be. And then if the bright young thing were up to it, he would come back 2 or 3 days later with the germ of an idea, and they would work on that together, and that would be the project. So just an astonishing man, again, just a very, very interesting character and a very original way of going about things. So here's first of all what Fred Griffith did. This is a picture of a blood agar plate. So these are colonies of bacteria, colonies specifically of pneumococcus, which are growing on this blood agar. The pneumococci are quite like blood. And the colonies on the left, those little sort of small pebbly things, are what are called R R stands for rough because the colonies look rough under a hand lens. And these pneumococci are completely harmless to mice. So you can take 1,000 million of these pneumococci, inject them into a living mouse. 24 hours later, the mouse is still scampering around, alive and well. On the right-hand side, you've got colonies which look a bit slimier. They're smooth, so S is smooth. That's the name of the strain of pneumococci. And these guys are lethal. With the R, the harmless ones, you could take 1,000 million, bang them into a mouse, nothing happens. With the smooth ones, you could take just a single pneumococcus, just one microbe, one germ, and inject that into a mouse. And 24 hours later, the mouse is dead and its blood is completely full of live S pneumococci. And what Fred Griffith was able to do in a series of completely illogical at the time experiments was to transfer virulence, lethality, if you like, from the R to the smooth, simply by mixing live R with an extract of dead S. So there was something that came out of the S, which was an inert chemical, that somehow got incorporated into the R and made them change their behavior. They then became the rampant murderers, that just a single one of which could kill a live mouse. And this extract of dead S proved to be quite a conundrum to isolate and to characterize. And it was called for a long time the transforming factor because it transformed these harmless R pneumococci into the smooth killers. And it was Oswald Avery, the man in New York, who actually 17 years later, with a series of co-workers, managed to prove that this was actually DNA. So this was the first demonstration that when it was known to be DNA, that you could transfer genetic material as DNA from one living organism to another. Admittedly, these are only bacteria, but later on, the same trick obviously was achieved with more complicated organisms. So this is a transfer of genetic material without any reproductive process involved at all. It's a passive uptake of a chemical. Now here's something else that Watson and Crick didn't do. You remember that A goes with T and C goes with G as the absolute central maxim to their structure for the double helix. Well, it was not their discovery. Again, if you read the book, and you're sort of left with a vague idea that they cracked it. Other people thought of it, but it was actually Watson and Crick that nailed it. Well, that's not quite true. The man that worked out that A goes with T and C goes with G is this remarkably cool dude here. This is Erwin Chargaff. He was professor of biochemistry at Columbia University in New York. Born originally in Lithuania, was fluent in about 4 or 5 languages. And towards the end of his life, he sort of lost, well, some people said he lost the plot. I don't think he did. I think he became cynical and he lost his ability to tolerate other people's inadequacies and foolishnesses. And he took against the biochemical establishment and wrote a series of really rude essays about the way that biochemistry and science were going down the pan. And he did this most fluently, apparently, in either German or Russian, but he also wrote some English versions as well. But in his younger days, Chargaff was a very noted researcher, and he was the first one to pick up the early technique of paper chromatography, and which had just been described for amino acids and proteins, for separating amino acids. And he worked out a way of adapting paper chromatography so as to be able to separate the bases in a sample of DNA and see what the relative proportions were. And when he started, the assumption was that actually DNA was a very dull molecule. It had these 4 bases, A, T, C, G, and it was just assumed that they were present in equal proportions. And there was a prevalent theory that, in fact, the molecule was just a string of repeating units of ATCG, 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 and these were called tetramers. So this was the tetramer hypothesis, that the DNA was just a very monotonous string of these 4 characters, end on end, going on forever. And what Chargaff did was to challenge that by actually doing some measurements, which nobody bothered to do before. So he got samples of DNA, And he came up with what are called Char-Gas rules. And he derived these by looking at the relative amounts of the bases in DNA from different species. And if you look first of all on the left there, this is DNA from calf, so just the calf thymus, the sweetbreads from the slaughterhouse. And that's a pie chart there with C, T, A, and G in it. And the first thing, if you look at it, it's obviously not divided into equal quadrants. It's not 25%, 25%, 25%, 25%. C is 20%, A is 30%, G is 20%, and T is 30%. So that's where the original supposition that C is somehow equated with G and that A is somehow equated with T, because the proportions of them, of those bases, those base pairs, if you like, are equal in the sample of DNA from the calf. Now, if you look at the right-hand side, this is a sample of DNA from a different species. This is actually from humans. And the source material here was 3/4 of a pint of human semen. So there are obviously occasions where you have to suffer for your science. And if you look at this, then again, you see that it's not 25, 25, 25, 25. And neither is it 20, 30, 20, 30. In this case, C is 15. So is G, that's 15 as well. A is 35 and T is also 35. So Char-Gas rules are basically that the base composition of DNA differs between species. So each species has got its own overall composition of DNA. And the other thing is that A goes with T and C goes with G. So those are Char-Gas rules. Now, the most important thing that Watson and Crick did not do was to deduce that DNA is a spiral or a helical molecule. And people had worked out that some molecules, for example, certain forms of sulfur, some early plastic molecules, the only way to explain how they could pack themselves into the right density and the right dimensions, they're able to work out the dimensions of molecules from viscosity and things like that. The only way that the atoms of the molecules could be packed to give all those data, make them consistent, would be if the molecule is a single strand, but it's not straight, it's actually bent into a spiral. And the first person to work out that DNA was a helical or a spiral molecule was Maurice Wilkins. And of all the people in the DNA story, I think he's the one who stuck it for the longest. He was a very, very methodical worker. He was a perfectionist. I think he was quite grumpy to work with, but the point is that he was a very systematic researcher. And for example, after the double helix structure had been published, he's the one who looked at it and said, well, actually, you know, the dimensions of the molecule are not quite right. So he spent the next 11 years actually working out exactly which bits of the molecule to crimp into shape to make it a proper, stable double helix. So the overall structure was right, but the dimensions of the various folds and the bends in the spiral were not quite right. So that gives you an idea of his methodical nature. And he came up with, the machine is sitting in front of there as an X-ray diffraction unit. And he worked out a way of getting X-ray diffraction pictures of so-called live DNA. And he was quite interested in this because he thought that if you could have live DNA as it operated in vivo in the cell, then it would probably be in the right disposition, the right structural format to serve as a living molecule rather than something dead that came out of a test tube. And he was quite right there because there were lots of preparations floating around at the time, including one by Char-Gas, which was very nearly pure, but not absolutely pure. And in there were fragments of DNA. So whenever you tried to work out the structure, for example, with X-ray diffraction, then you ended up looking at a whole mishmash of things of different lengths of DNA, some of which had been broken up in the extraction process. So you could end up with something that wasn't terribly informative. And what Maurice Wilkins came up with was the notion of looking at squid sperm and cephalopods like squid and cuttlefish. During the mating season, the male cruises around with a little bag of spermatozoa called the spermatophore, and he has it tucked away in some nook and cranny of his anatomy, and when he reaches the female of his desire, he implants the spermatophore in a bit of her anatomy, and away you go. Now, inside the spermatophore, the spermatozoa are lined up. They look in section like a sort of little array of torpedoes ready to fire. It's quite a miraculous sight. And what Maurice Wilkins worked out was a way of rupturing the spermatophore very gently so that the spermatozoa still remained in the right orientation. And then amazingly, he was able to make a preparation of that and get an X-ray picture of it. And this is his X-ray picture of, not quite living, but ex vivo squid sperm. And what you see there is a bold black capital X in the middle. And again, I won't go into the crystallography because I can't claim to understand it. But if you have a regular spiral, a regular helix, and the X-rays are shooting up it and bouncing off it, then at a certain angle, you end up with this X shape. And Maurice Wilkins was fascinated by this because when you put any other kind of crystal in front of an X-ray and look to see how the X-rays are scattered, then you end up with a geometric array behind the crystal. You can pick it up on a photographic plate, which looks like a symmetrical picture of dots. But the point is that the dots from any other crystal are distributed over the whole of the field. And if you look at this picture, then you'll see that you've got the X, the arms of the X are filled in, as it were, but the areas between the arms are left blank. And Maurice Wilkins deduced that this was indeed a spiral, and that you've got these voids between the arms of the X because this is where the X-rays are effectively looking up the middle of a hollow structure. So there's very little, the base is not particularly X-ray bouncy, so you don't see very much there. The main image you get is actually off the sugar phosphate. The phosphate's heavy, so it scatters X-rays. So this X basically tells you that the molecule you're looking at is spiral. So this was the first evidence that DNA was actually a spiral molecule. The other thing that Watson and Crick didn't do was any experiments whatsoever on DNA. They never touched the stuff in the lab. And it's interesting reading the end of their paper because there's the beginning, which you've already seen, so they're staking their claim. And at the end, towards the end of that paper, again, it's a very short paper, there are no data in it. There's not a single number in it anywhere. And the reason for that is that they didn't actually do any of the experiments themselves. Towards the end of the paper, they say, we were not aware of the results when we devised our structure, which rests mainly, though not entirely, on unpublished experimental data and stereochemical arguments. Well, let's look at that sentence. We were not aware of the results. Well, the results were those published in the other two papers in Nature, right beside this Watson and Crick paper, the famous one. As I mentioned, there were two other papers in the same issue of Nature about DNA. One was by Maurice Wilkins, the other one was by Rosalind Franklin. And when Watson and Crick say, we were not aware of the results of these other two papers, they are lying, because actually they had knowledge. They managed to, you could argue that it was not a terribly deceitful thing to do, but they managed to get privileged access to something that wasn't circulated to them, which contained the data which Rosalind Franklin generated about some of the sizes in the spiral, which made them then work out how those steps of the two base pairs actually fitted into the structure. And when they say at the end of this sentence, our structure rests mainly, though not entirely, on unpublished experimental data and stereochemical arguments, that's actually just fluff to cover up the fact that they nicked other people's data to suit their purposes and develop their own structure. So this is the so-called Dark Lady of DNA. This is Rosalind Franklin. Again, one of the great enigmas of 20th century science, ferociously bright, ferociously prickly, very difficult to work with. She and Maurice Wilkins sadly fell out very quickly indeed. I think reading his papers and his letters, he did his best to try and get on with her, but she had a very difficult personality. On her side of the argument, she found him intolerable. But I think he was actually a gentleman. He was just uncertain as to how to deal with a very strong-willed woman working alongside him. So Rosalind Franklin, again, she was a professional X-ray crystallographer, if you like. She had done that in Paris before she moved to the unit at King's, where she worked alongside Maurice Wilkins. And in Paris, she tried to work out the X-ray structure of clays and various sort of random molecules. And clays don't form neat, pretty crystals, which are easier to analyze through X-ray crystallography. They're sort of semi-organized. And she'd managed to make great headway in sorting their structures out. So when she came to look at DNA, she already had the background, not only of X-ray crystallography, but also of dealing with these non-crystalline, partially disorganized, partially random structures. So she was definitely the woman who was going to make great strides in this area. And she did. And you were probably well aware of the famous photograph 51, which was taken in 1952. It was actually taken after Maurice Wilkins' squid sperm X-ray picture. But photograph 51 shows the same big, black, bold capital X in the middle, which tells us that the structure she's looking at is helical. She did a very systematic series of studies looking at DNA in various forms. This is the wet form of DNA, so there's quite a lot of water in the structure. So it forms this nice helix, and you get that lovely X pattern. And she also showed that if you have slightly drier DNA, then you end up with a picture that doesn't look like that at all. It looks more like a crystal. And the reason for that is that when you pull the water out of the structure, the helix kinks in different ways and actually ends up looking more like a crystal than a spiral. So she managed to sort all that out. But in so doing, she fell out not only with Maurice Wilkins, but she also fell out with the head of the unit at King's, where they were both working, who was a man called John Randall, who comes in at the very end of the story. And the upshot is that she had to abandon her, she had basically was thrown out of the lab. She had to abandon her DNA study. She did write up the papers and publish them with a man called Ray Gosling. And she then went on to work at Birkbeck College on the structure of viruses. And in fact, her most enduring, her most beautiful work was actually on the X-ray crystallographic structure of tobacco mosaic virus, and she worked on the polio virus and lots of others as well. And you probably know that she got ovarian cancer and died at a very young age, just 37, and was greatly lamented. She was particularly lamented actually after Jim Watson's book, The Double Helix, came out, because she was portrayed horribly as a nasty, difficult, impossible, not very bright, English blue stocking, unapproachable woman. And again, aspects of that might have been founded in some way on her character, which was not easy, but it was horribly embroidered, gratuitously embroidered to make the story a good read, because it needed a villain. There wasn't any sex or violence in it, so Rosalind Franklin, or Rosie, as Watson very deprecatingly called her, was the nearest thing to a villain in the story, so that's what she became. Her epitaph is quite interesting. It says, Rosalind Franklin, scientist, and her contributions to virology will be of lasting benefit to mankind. And we don't know who wrote the epitaph, but she had enough time before she died to know what would be in it. So I'm pretty sure that if she didn't write it herself, she approved of it. And it's interesting that in there she mentions her work on viruses is specifically singled out, but there's no mention whatsoever of her work on DNA. And again, she, I think, regarded this just as a project, much as her clay work in Paris had been a project. It was a phase in her life. Same for DNA. She did it, she moved on, and she went on to viruses where she really made her mark. So this brings us to the outcome of the story of the double helix, if you like. And this is the Nobel Prize ceremony in Stockholm in 1962. And reading from left to right, you've got Maurice Wilkins. So he actually did end up sharing the Nobel Prize with Francis Crick, who is third from the left, and with Jim Watson, who is second from the right. So that's the triumvirate, if you like. Rosalind Franklin had died by then. There was a lot of fuss about whether she deserved a share. In fact, operationally, it would have been difficult because at that time we weren't supposed to have more than 3 people sharing the prize. But the other thing, I think if you look dispassionately at what she did and what the others did, then she did a lot, but she didn't do quite as much as the other 3. Just for completeness sake, the other 2 people in the picture, sorry, the other 3 people in the picture, you've got Max Perutz, second from the left, and John Kendrew on the right there. They got the Nobel Prize for chemistry that year for discovering the structure of myoglobin and hemoglobin. And in the middle there between Crick and Watson is John Steinbeck, who won the prize for literature. So just to finish off with, I'd just like to tell you about 3 of the lost heroes. And again, the people I've mentioned up to now are people that I'd heard of, and quite probably you have as well. I'm not so sure you'll have heard of any of these 3 though. The first thing I'll do is just show you this picture. And from what you know now, you will automatically say, well, that is a big, bold, black capital X. That is the shape of a helical molecule. That's an X-ray diffraction picture. That's okay. So it looks just like photograph 51. However, if you look at it in the full, it's not called photograph 51. It's called B299. And if you look at the date, it's been trimmed off a little bit, but that's actually the 1st of June, 1951. So this picture was taken a whole year before photograph 51 was taken. So who on earth did that? Well, it wasn't anybody working at the unit in King's where Rosalind Franklin and Maurice Wilkins worked. It was taken by a man working in Leeds with an amazing guy called Bill Astbury. And the man who took the photograph was called Elwyn Bayton. And Bill Astbury, his boss, was a professor of textile physics up in Leeds. He had trained with Bragg and various people. He was an expert X-ray crystallographer, and he's the one who managed to get X-ray pictures of various natural fibers, and also one of the hairs from the head of Wolfgang Amadeus Mozart. And Elwyn Bayton was working alongside him as a PhD student and was supposed to be doing something with bacterial flagellae, which is what Astbury was interested in at the time, how those are put together. That work didn't go terribly well. A sample of DNA was sent to the lab by somebody who wanted to know what it looked like under the X-ray camera. And Bayton said, well, I'll do that. So he did it, and this is what he got. And this is essentially wet DNA, just exactly as Rosalind Franklin had photographed it. And it's the same general picture as Maurice Wilkins had got from his squid spermatozoa. And it's a perfect picture of a helix. And what happened to it? Nothing. Bill Astbury, brilliant man that he was, looked at it and could make head nor tail of it. And again, he was used to looking at patterns, symmetrical patterns of spots. So this thing, where there were huge voids between the arms of the X, he had no idea what it meant. So he said, I'm not going to try and get a grip to this. Let's go back to the bacterial flagellae. And that's exactly what happened. So that picture was never even published. It lay around in the Astbury papers and the special collections of the Museum at Leeds. You can still see it there today. And it was only many years later that its significance was appreciated. So Elwyn Bayton actually was one of the first people to get a laboratory specimen of wet DNA and to get that capital X picture. And that was before Rosalind Franklin did it. So my second lost hero is a man who worked out how the sugar phosphate of DNA might be stuck onto the base. And what he's done here is he's worked out the structure of just a single strand, if you like. It's the sugar phosphate bit. I'll show you the sugars in just a moment so you can see how the organization is. And he's just got a single base attached to it in what he thinks is the ideal conformation. So if you like, if you remember the double staircase with the 2 halves of the stair being split and the 2 handrails moving apart, this would be one handrail with half a step attached all the way down. But in this case, for the experiment, it was just a single base. So these are, if you like, these are A's all the way down. And what he's done here is he's drawn the pentose sugar, so it's a pentagon. That's the deoxyribose sugar there. And if you look at the shape that those pentagons describe, you can see that they're actually rotating. So this structure has actually got a spiral, a gentle spiral twist to it. And if you look at the top there, you'll see that there's essentially a horizontal line going across with circles in it. That is the base. And the bases, you remember when we looked at them from above, they were either a hexagon or a hexagon bolted onto a pentagon. When you look at them from the side, they're actually completely flat molecules. So they're planar. So what we're looking at here is we're looking across the plane of the steps. So we're seeing the steps end on, and that's why they look like a single line. So this is a spiral to which the bases are attached. So that's one of the handrails. So again, that's quite a long time before Watson and Crick came up with their idea in 1953, because this was 1949. And the man who did that was a Swedish worker called Sven Furberg, who was working at Birkbeck College with J.D. Bernal, who was the great crystallographic king at the time. And what happened to this? Well, nothing. J.D. Bernal was a great lover of the communists. He was very politically active. And towards the end of this man's PhD, Bernal was in Russia quite a lot of the time, talking at the peace conferences just before the Russians exploded their plutonium bomb and so on. So Sven Furberg wrote up his thesis himself. He published it in the little addendum when he got back to Stockholm, and that's as far as it went. So this structure, which could actually have made a big difference to the debate, essentially disappeared back to Sweden and was published in an obscure kind of way, and Furberg didn't get the recognition that he deserved. And my third lost hero is the man that drew this very scrappy diagram in the margin of his PhD thesis. And what he's drawn here is actually a double chain of DNA. And it's not, as we would imagine it today, as two continuous strands with crosslinks. What he's imagined is that it looks rather like a broken ladder. So some of the rungs, if you like, go across both sides of the ladder, and then there are gaps. And the way that he worked out the structure of DNA was from the way that it behaved when it was being subjected to flow in solution. And he worked out that the DNA came in two chains which were straight and broken, but they were actually held together by hydrogen bonds. And the hydrogen bonds existed between, they pulled together bases in alternate sides of the chain. So if you look at the little section in the middle there, on the top you would have a pyrimidine, and on the bottom you would have a purine. So this is essentially an example of A going with T and C going with G. So in this structure, you've got a double strand, you've got base pairing held together by hydrogen bonds, exactly as Watson and Crick have done it. The thing that is wrong is that it's not a spiral and it's not complete. Now, the man that came up with this was in 1947, so several years before Watson and Crick. And this is a man called Michael Creeth, who actually worked in Bristol latterly. And he did this work as part of a PhD. His PhD supervisor was a man called Gunnar Masson, who worked in Sheffield, but was a Scot from Edinburgh, desperately wanted to be the professor of biochemistry in Edinburgh, failed in that, resigned his post in Sheffield, so he left this PhD student in the lurch, and then he went back to Edinburgh for a meeting and got killed in a train crash on the way back to King's Cross. So this poor guy had to write up his PhD in a hurry, which is why this beautiful diagram is just a scribbled note in the margin of his thesis. And this work, again, was never presented or published. So I'll leave it to you to imagine what might have come about if these three bright young men, these three lost heroes, had actually been put in a room together and said, okay, what do you think DNA really looks like? because one of you's got a double strand, one of you's got a spiral in it, one of you's got a distinct spiral from X-ray crystallography, and one of you thinks that you've got base pairs linked across between the strands using hydrogen bonds. So just going back to this picture, again, this is Watson and Crick in their full glory. But as I've indicated, there are other people in the story. That's Friedrich Miescher, who discovered the stuff towards the end of his life when he was just about to get TB and head off to the sanatorium. Fred Griffith, very sad ending to him. He lived in Pimlico, in a big square just south of Victoria. And when the Blitz started, he said, I'm not going to move out for any German. And he and his best friend were in the house. They went up to do some fire watching from the roof, and they took a direct hit. So they dug the dog out of the ruins, but not him. Avery, again, very interesting, charismatic man who actually kept that photograph of Griffith on his desk when he discovered how Griffith had come to a sticky end, and took it with him after he retired from the Rockefeller. Rosalind Franklin, again, the dark lady of DNA, the iron lady of DNA, if you like, and Maurice Wilkins, who to my mind is the man that actually contributed, I think, the most to working out what the structure was. And I'm going to finish with the man that you haven't met yet. I've mentioned him in passing, and this is John Randall, looking very dapper here. Again, one of the founders of biophysics, if you like, very, very bright man indeed. A technological war hero because he was the discoverer of the cavity magnetron, which basically allowed the Brits to micro-miniaturize a very powerful radar set and put it inside fighter aircraft and anti-aircraft guns and things like that. So that was his invention with a man called Harry Boot. After the war, he came down, set up the biophysics unit at King's, and that's where he recruited, first of all, Maurice Wilkins as his right-hand man, and then Rosalind Franklin. And when Rosalind Franklin was recruited, he said, you can come and work on proteins in solution with X-ray crystallography. And she said yes. And then he sent a follow-up letter saying, we've had a change of heart, and we would like you to work on DNA. I've discussed it with all the senior people in the lab, and we're all happy. So DNA will be your project. You'll be the only one working on it, and you've got a PhD student to help you. So she was delighted. It's very unfortunate that when John Randall sent that letter to Rosalind Franklin, Maurice Wilkins was on holiday with his girlfriend, because the senior person working on DNA in Randall's unit was none other than Maurice Wilkins. And when he came back, he was aware that Rosalind Franklin was there and was claiming ownership of the PhD student, but he was too timid to say anything about it. And it was only later, when he was writing John Randall's obituary for the Royal Society, that he discovered this letter in John Randall's effects. He'd kept a copy of it. And he was very, very upset indeed, because he realized that John Randall had invited Rosalind Franklin in to poach his project effectively, and ultimately to set him against her. If that hadn't happened, then they might have collaborated, and the double helix might have been the Wilkins-Franklin model of DNA instead. So I'm just finishing with words from, oh, right. Well, it seems to have jammed. Okay, well, my last slide, I'm sorry, was a quote from John Masefield. It was from one of his sonnets, and it talks about a thing of restless cells and salt and water. And that was something that John Randall had heard an American friend lecturing about, and he wanted to know where the quotation came from. So this was another letter that was in John Randall's personal effects when he died. And it shows that John Randall had a great sense of poetry and a love of poetry as well. And of course, you can't make restless cells with just salt and water. You need a double twist of DNA as well. Thank you very much.
Gareth, thank you. The technology prevents the audience breaking out in applause. But I have to say that was the most interesting biochemistry lecture I've heard in a long time. And I'm not saying that to make you feel good. Timothy Chambers is applauding, actually. Well done, Tim. Thank you. And so is Peter Carpenter. So actually, clearly we can break out in applause, although nobody hears it. So, folk, there may well be some questions. Thank you, Gordon Sturrett, for your applause. And Charles Ward, they're all going to do it now. What I'd like to do is to give people a chance to ask some questions, but maybe to keep it orderly. Could you just write your name in the chat, which I'm going to watch? And if you pop up in there, then I'll come to you so that it's not chaotic. And I have a question, Gareth, for you, but I was just thinking about your Watson and Crick and Franklin and how, for various reasons, she was excluded. And it reminded me of that factual story from reproductive medicine where what's their names, Steptoe and Edwards published the test tube baby story.
Yeah.
And they forgot completely in all the hoo-ha about a woman called Jean Purdy, who was the scientist who'd done lots of the work, and it was a great source of sadness, really, that she got excluded from that process. So before people come in with some serious questions, I've got a slightly unserious question for you. So Chargaff, your Lithuanian multilingualist guy, managed to get, you said, 3/4 of a pint of semen. So that's over 400 mils of semen.
It was a team effort.
Clearly, very big team, because that would have meant 80 men producing semen.
Again, the details are
lost in history.
Lost in history, but yes, it was definitely a team effort.
Wow. It doesn't bear belief. Okay, so serious questions now are permitted. Anybody who'd like to ask a serious question. So somebody's being very rude. This feels like an account of a lot of people with high ability autism. I think that's probably a real question, actually, is it? So I think that's Peter Carpenter, I think, and he's saying that this feels like an account to a lot of people with high ability autism who just couldn't work with each other. Is that a fair comment?
I don't know. I'm sure Peter's worked in a lab as well, but I don't recall any of the places I've worked at the bench where everybody has been completely harmonious. Mm. And you probably need a competitive edge to keep people pushing forward in the right sort of general direction. I think certainly Rosalind Franklin, I don't think I've been unfair to her. Although interestingly, I have met people who worked in the same unit. Tony North and his wife Margaret, they are now up in Leeds. And Margaret was working alongside Rosalind Franklin, not in the same project, but similar, and discovered that she was going up to, they were both going to Shropshire for the weekend. So they arranged to meet up, and Margaret said she had a lovely walk. They climbed Carrick together. They had a great time, and she was lovely company. And I think that this is just the impact of working in a high pressure environment. I think people don't behave very well, but certainly the pressure is on them. They find it very difficult to relax.
Thank you. So there's a dearth of input on the questions. Tim, would you like to ask a question? Do you want to unmute yourself first? Thank you.
Thank you, Gareth. That was, I learned a lot. Thank you very much. Has there been any suggestion that anti-Semitism played a part in Rosalind Franklin's ostracization? Or?
No. She came from a Jewish family, but she wasn't devout. She didn't make a big thing of her religion. Her sister is a younger sister still alive, Jennifer Glynn, who's Ian Glynn's wife. He's the man that discovered sodium potassium ATPase up in Cambridge. And I've talked to her at length about, in fact, she's written an excellent book called Revealingly My Sister Rosalind Franklin. And the family were Jewish, but it wasn't a big part of her life. And I don't think that anti-Semitism came into it at all. No, I think it was more just a difficult personality.
Thank you.
Thanks. Victor Barley, you'd like to ask a question. So if Victor, you could unmute and turn on your video.
Yes.
Oh, there you are.
I was under the impression that Rosalind Franklin and her team were working with the possibility of 3 strands to the helix rather than 2, and that rather misled them. Is that correct?
Well, it was actually somebody on Maurice Wilkins' side who came up with the triple helix, and they looked at it and decided that it simply wouldn't work. And then shortly after that, Linus Pauling, who was already a Nobel Prize winner for chemistry, rewrote a lot of 20th century chemists, so a very, very serious player. He actually published a structure which had 3 strands, and it was crazy. And nobody quite worked out, at least it's crazy to professional biochemists and structural chemists. And this structure was so bizarre that it would have exploded, because it was inside out and all the forces were wrong in the core of the structure. So it could never have held together, it would have blown itself apart. Maurice Wilkins actually came up briefly with a structure with 4. This looked like sort of a double snake serpent's physician's staff, Asclepius type thing, but 4 of them intertwined, and then he realized that wasn't going to be any good. So yeah, 4, 3, and then 2.
Thanks, Victor. Well, perhaps Gareth, while we're waiting, you could play the flute. Or whatever. What do you play?
Yeah, flute. Yeah,
it is the flute. Yeah. Perhaps not. Well, if people aren't rushing to ask questions, I'm not going to keep Gareth any longer. I think you've treated us to a learned discourse on the story behind DNA tonight. It's very evident from what you've said in response to questions and in response and in the entire talk about how much work you've put into this and the amount of time and effort that it's taken. It's very gratifying then to hear it all. Gordon, is that you wanted to ask a question? Just thank you. Great. So we're going to let you go. We greatly appreciate you giving your time and the effort required to put this together. I think we've benefited greatly from it. As Tim said, we've learned a lot. So I certainly have had my spheres widened, even though I've read your book, but I didn't quite grasp everything that you've put together. So hearing it all kind of packaged neatly, that was very helpful. And Gareth's book is available in all good bookshops and online. Unravelling the DNA Helix. So thank you very much, everybody. Thank you very much, Gareth, for joining us. And I'm sorry we couldn't have had you in person. It would have been really good. And yes, indeed.
Thank you. Thank you very much. It's been great.
Thanks, Gareth. Good night, everybody. We'll see you all in January for the next meeting. Meanwhile, keep well and keep safe and have a lovely Christmas. Bye-bye.
Bye. Thanks.